So we talked about human meiosis. I want to show you that here's an example of an organism, this is a plant, that uses a completely different life cycle than humans do. So in plants, the main organism is diploid, and they undergo meiosis just like we do. But when they undergo meiosis, when plants undergo meiosis, they produce spores. So meiosis produces spores, then spores grow up into an organism via mitosis that's also haploid, and that haploid organism makes sperm and egg via mitosis. And mitosis then makes sperm and egg, they fuse together to make a zygote. So this is known as the alternation of generations, which you'll learn more about in Bio 2. But what I want you to see here is that not all organisms have the same life cycle. Some organisms produce gametes by mitosis instead of meiosis, and they make spores by meiosis. A little bit different than we're used to seeing. Okay, so let's look at overall what meiosis does. What meiosis does is it produces cells. It usually makes 4 cells. So let me start there. Let me grab my pen. Meiosis makes four haploid cells. Let me modify that. It makes 4 unique haploid cells. So ultimately, those 4 cells are probably not similar to each other. They're a little bit different from one another. So let's start here. Here we have a diploid cell, 2n equals 2, right? There's 2 chromosomes in it. Not a male, but a maternal and a paternal chromosome we can see there. Those chromosomes duplicate in the S phase of the cell cycle. And so now we have replicated chromosomes there, or duplicated chromosomes there. And then meiosis 1 kicks in. And look what happens at the end of meiosis 1. At the end of meiosis 1, you have 2 cells produced. These are haploid. These are haploid cells, but they consist of duplicated chromosomes, right? So the chromosomes look like this. There's 1 chromosome in each of those cells. Up here we had 2n of 2. Here we have n equals 1 in these cells down here. Then meiosis 1 kicks in, and in meiosis 1, these duplicated chromosomes separate, and we make a total of 4 cells, each with 1 chromatid each, 1 chromosome each, consisting of 1 chromatid each, and so n equals 1. So here we have n equals 1, but chromosomes consist of 2 chromatids. Here we have n equals 1, but each chromosome is 1 chromatid. So compare this cell here, for example, to the one we started with. It's almost the same, right? Except it's 1 chromosome instead of 2. That's why it's haploid, or N. Now, the phases. We're going to walk through these kind of quickly in the video, and then I'll put up another video where I try to draw them for you, okay? Here we have a cell, 1, 2, 3, 4, 5, 6. So 2n equals 6, it looks like. And this is going to look, there's going to be a lot of similarities, but also a lot of differences, especially in meiosis 1, will be a little bit different than mitosis. So first of all, when we number the phases here, we have to number the phases here. So it's not just prophase, it's prophase 1. That's very important to know which number you're dealing with. The first thing that happens here in prophase 1 is that homologous chromosomes pair up. They are in pairs. They're like touching like that. Homologous chromosomes pair up. This is a process known as synapsis. Okay. Now, non-sister chromatids, shown here, are touching, and that's what that chiasmata word means. It means that the parts are crossed over. And that's where crossing over will occur, at the chiasmata. That's where the arms of non-homologous chromosomes are touching. The spindle is forming, the nuclear envelope is going away. So we have no pro-metaphase in meiosis. It's prophase and metaphase 1. Okay, so the nuclear envelope goes away, the spindle's forming, the chromosomes are condensed, but they're touching here. We call this a tetrad when they're formed up like this in synapsis. Synapsis is the verb, that's how they get there. And the tetrad is simply the structure they form, which it's called a tetrad because there are 4 chromatids involved. Okay, then metaphase 1. The tetrads line up at the metaphase plate. Anaphase 1, tetrads separate, right? So homologous chromosomes separate, and some of them go up and some of them go down. Now, during this prophase and metaphase, crossing over has occurred, so there are pieces of this paternal chromosome stuck to this maternal chromosome and vice versa. So crossing over has occurred, something we had not seen before. Then at the end of meiosis 1, telekinesis, cytokinesis kicks in, telophase kicks in, we form 2 new cells. Now those 2 cells are both going to go into prophase 2 next. So what happens in meiosis 1? Chromosomes condense, homologous chromosomes pair up. That's a process known as synapsis, and that forms that structure I mentioned a second ago called a tetrad, which we see below here on this image. Crossing over occurs where non-sister chromatids touch at an area called the chiasmata. The spindle forms, the nuclear envelope breaks down, all those things we looked at that took place in prophase and metaphase, and prometaphase, I should say, of mitosis. Metaphase 1. Pairs of homologous chromosomes are arranged at the metaphase plate. Each homologue, so each homologous chromosome, is attached to microtubules from the opposite pole. We'll draw that here in a little while. And then we go into anaphase 1, where homologues move to opposite poles, and telophase 1. Each half, oh, I just threw my pen on the floor. Each half of the cell has a haploid set of chromosomes, but those haploid chromosomes consist of 2 sister chromatids. So what do I mean by that? Let me see if I can draw like this. All right, so the chromosomes still look like that. Okay. So homologues separate up here. So here's one, one's going up, one's going down, right? That's what we mean by homologues move towards opposite poles. So instead of this, like we had in mitosis and anaphase, in anaphase 1, we have this happening. I call it the Richard Nixon. I am not a crook. They separate like so. Cytokinesis kicks in. Then we enter into prophase number 2. And here chromosomes condense, nuclear envelope breaks down, spindle reforms, same thing. But this one's going to look a lot more like mitosis than the last one. Things hook up to the spindle. Here in metaphase 2, the homologous chromosomes are lined up at the metaphase plate. Now they're drawing the cell up and down instead of, or they're oriented it differently as you, they orient it differently so you can see it there. They're going to move left and right instead of up and down in the drawing, but they're lined up at the metaphase plate. Anaphase 2, homologous chromosomes separate. I shouldn't say, let me rephrase that. Anaphase 2, sister chromatids separate, but you can see that they're not identical anymore because crossing over has changed some of their genetic composition. So remember, we're looking at 2 different cells here that are doing this. And then telophase and cytokinesis, the nuclear envelope reforms, chromosomes decondense back to chromatin. And when we're done with this, we have 4 cells that have been produced. So meiosis 2, prophase 2, spindle forms. Same stuff we've talked about in mitosis happens. Metaphase 2, chromosomes positioned at the metaphase plate. Kinetochores of microtubules are attached to opposite poles. So what's going to happen next is they're going to separate sister chromatids, and we're going to form daughter chromosomes. But in this case, they won't necessarily be twin daughters, right? Anaphase 2, sister chromatids separate and move towards opposite poles. Telophase 2, the nuclei reforms, chromosomes decondense, cytokinesis kicks in, we form new cells. Stuff you're familiar with. Those cells are now basically going to mature into an egg or a sperm or a spore if it's a plant. So 4 cells are produced, 4 daughter cells are produced, and they are all genetically different from one another. That's a cool part. Okay.